we will be talking about plasma profiling.
Hello, everyone. My name is Xiao-Jun Ma. I'm the Chief Technology Officer at Alamar Biosciences. First, I'd like to thank the organizers for the opportunity to speak here. I will be talking about using the NULISA technology to profile the plasma proteome to identify biomarkers of aging. So what is NULISA? NULISA stands for Nucleic Acid-Linked Immuno-Sandwhich Assay. It is an immunoassay that was designed from ground up to have both ultra-high sensitivity and high multiplexing capability. And shown here is a schematic of the molecular design of the assay. It is an immuno-sandwich assay, so it uses 2 antibodies per protein. And it is also a proximity ligation assay. So it has an oligo attached to each antibody. When the antibody pair binds to its target, it brings the 2 oligos together, which can be ligated to generate a reporter molecule for quantification. What differentiates NULISA from traditional proximity ligation assay is that we have built in 2 different mechanisms to purify the immunocomplex before we perform proximity ligation. One is through the poly A sequence that's attached to the capture antibody shown in green, and the other is through a biotinolated oligo that's attached to the detection antibody shown in blue. Here's the actual workflow. We first form immunocomplexes in solution by incubating the antibodies with the sample, and we capture the immunocomplex onto the first bead oligo DT beads through oligo DT poly A hybridization. And we wash away all the unbound material, and then we actually release the immunocomplex from the oligo DT beads by incubating the beads in a low salt buffer and recapture the immunocomplex onto a second bead, streptodaptin beads, which binds to the biotin molecule attached to detection antibody. So after washing, we're left with essentially highly purified, fully formed immunocomplexes on the streptodaptin beads, which we then perform proximity ligation to generate a reporter molecule. This double capture release mechanism really reduces the assay background from traditional proximity ligation by over 10,000-fold to achieve attomolar level of detection sensitivity. For multiplex detection, each reporter molecule also contains barcodes for the target, so we can detect hundreds to thousands of target proteins in a single reaction. All of these steps were carried out in a fully automated system called Argo, as in the picture shown on the top right. The Argo instrument takes the sample and generates a DNA reporter library that's ready for NGS sequencing. To leverage the ultra-high sensitivity and multiplexing capability of NULISA, we developed the information panel. The information panel now includes 250 proteins listed here. As you can see here, these are mostly cytokines and chemokines and their receptors. This panel, we believe, has the most complete coverage of chemokines and cytokines in a single panel on the market today. With the sensitivity and multiplexing capability, we can measure all of these 250 proteins in a single reaction using 10 microliters of sample volume, with attomolar sensitivity and also very high level of precision with both interplate assay CV with less than 10%. NULISA also has incorporated a mechanism to expand the dynamic range of the measurements, so we can combine both high and low abundance of proteins in a single reaction without diluting the sample. In this panel, we actually covered about 12 log of dynamic range. The second panel we developed is the CNS disease panel, and it has 120 proteins shown here. This panel was designed with inputs from multiple pharma customers and academic KOLs. As you can see here, it includes many of the key biomarkers of neurodegenerative diseases, such as amyloid beta fragments, GFAP, NFL, and multiple forms of phosphorylated tau proteins, and many others. Again, with the multiplexing capability and the sensitivity of NULISA, we can measure all of these 120 proteins in a single reaction using just 10 microns volume with very high detectability and 98% detectability in plasma and 88% in the CSF. So in a collaboration with the Methuselah Foundation, we apply both panels to profile a aging cohort shown here. And this cohort has a even split between male and female, and also uniform distribution across different age groups, has quite a diverse race and ethnic background. With the data, we used the elastic net to select biomarkers to predict chronological age. We performed 50 rounds of cross-validation and identified 210 age-related proteins. We rank these proteins by their frequency of selection across the 50 models. And as shown in the red dot, we demonstrate that the top most frequent 170 proteins were able to build a model, generate the lowest prediction error. We applied the selected model to a test set. As shown on the right, we're able to predict age with a very high correlation with chronological age, with correlation coefficient of 0.93. We mapped the 210 protein markers we identified in protein-protein interaction networks. And the first we notice is a highly connected chemokine network with CCL11 showing red at the center of this network. CCL11 is a well-known chemokine that increases with age, and this is consistent with the previously described aging mechanism, with inflammation being a key driver of the biological aging process. I also like to highlight a second protein shown in blue called KLRK1. This protein actually decreases with aging. This is a protein that has not previously been characterized to be associated with aging. However, it functions as a receptor on NK cells, and it activates NK cells to eliminate senescent and dead cells in the body. So a decrease of this protein with age definitely could contribute to the process of biological aging. We compared our age signature to another signature that was recently published in Nature Medicine by Argentieri, and we found there's a limited number of proteins were in common between these two signatures, but these two signatures are largely unique to each other, indicating that we may capture both shared and also distinct mechanisms of aging. To summarize, we have profiled a total of 322 inflammation and neurodegeneration related proteins. We identified many known as well as novel age-related proteins. We demonstrated that accurate age prediction can be achieved with less than 200 proteins, and the protein content of the signature can provide insights into the mechanisms of biological aging. We envision that a compact multiplex panel that focuses on hallmarks of biological aging could be a powerful tool for the aging research community. With that, I'd like to thank our collaborators from the Methuselah Foundation and also our teams at Alamar Biosciences. Thank you.
Thank you. Thank you so much. Actually, Xiao-Jun was amazingly good with time, so we have about a minute. If there is any pressing question, I mean, maybe we can address it right now. Otherwise, Chiara gets an extra minute. Oh, we have a question from Nir.
That's terrific. I just didn't understand in the technology why it's better than other autoimmune assays. You said that you're removing something. How does it make it better?
So the fundamental insight in building NULISA is that we believe the really truly improve sensitivity, you need to reduce the background. You cannot just increase signal. So that's why we built in this double capture release mechanism that really suppresses background from non-specific binding and cross interactions. Yeah.
Great. If you have any other questions, later during the break. Thank you so much. Thanks.